Hello, everyone. We're just letting everyone in. In the meanwhile, let me thank you for coming to this webinar. This is the third webinar of EARA webinar series. So the first one was on patient engagement. The second one was on the role of veterinarians in animal research. And this one is about new approach methodologies. So a discussion on how new approach methodologies and animal research can and should complement each other. So this will be recorded. So you probably saw that it's being recorded in a warning in your Teams. Just to briefly make a notice on our social media campaign. It's called Be Open About Animal Research Day. We do it every year. This year will be on July 9th. It will be during Fans Forum. So we will be doing this in that setting. And we will, for the first time ever, we will have a thematic approach to this social media campaign where we ask institutions to contribute with materials on videos, written statements on how institutions are making efforts to become more transparent about animal research. And this year, we have the theme of NAMs and animal studies, not a competition, a collaboration. So if you and your institution are interested in participating, please check this QR code or just go to our website for more information. So this is the schedule for today. We will have 5 speakers. We'll have an introductory speech by Kirk Leach, the Executive Director of EARA. Then we'll go to Magdalena Loreno-Vix, hope I'm not butchering your name, from the PPRC, Miguel Prudencio from GIM in Portugal, Walter Westerink from Charles River Laboratories in the Netherlands, Sonia Beckham from the Federal Agency for Medicines and Health Products in Belgium, and then we'll start the panel discussion. So the webinar rules are pretty simple. So every time you have a question for the speakers during their talks, you can do it in the Q&A. This will not be addressed until the panel discussion, but they will be there and we will select them for hopefully the speakers to approach them in the panel. So speakers can also reply during the panel discussion or in the Q&A itself. And you can also use the Q&A during the panel discussion to write down questions during that time. As I said, the webinar is being recorded and an edited version will be made available soon on our YouTube channel. So without further ado, I would like to introduce Kirk Leach, the Executive Director of EARA, to make an introduction on EARA's view on NAM's integration on biomedical research and how animal studies play a role in this integration. So the stage is yours, Kirk.
Thank you, Nuno. Yes, so I'm going to say just a few words about EARA and why we're holding this webinar today. Next slide, Nuno. So for those who don't know, EARA is, I think, probably the only really global advocacy organization. I'll probably get tired of saying this at some point, but I still think we're a relatively new organization created in 2014. We now have over 240 members across 47 countries. In Europe, we have offices in Brussels, London, and Lisbon. That's the global map of EARA. As you can see, Europe is still, I think, our most important kind of geographical area. But as you can see, there's now a spread of EARA members across the globe, and we welcome interest in EARA from outside of Europe, obviously. They're the kind of benefits of EARA membership. As Nuno said, you will get these slides after the presentation in the kind of the recording. But, you know, EARA, I think, is a trusted voice for biomedical research. Membership of EARA will give you great advice on openness and transparency and practical training on communicating NAMs or animal models, and through our newsletters and other materials, you will get a real insight into significant developments in policy and regulation. We have a weekly news digest, which you will probably get from registering for this event, but also there's a policy briefing, which looks at significant policy and regulatory changes in Europe and wider. So if that's of interest to you, QR code will come up in a minute. You can either wait for this or use your camera and get it. So why this webinar? I think for EARA, NAMS, New Approach Methodologies, is one of the defining issues of life science at the moment. And I think how the research community responds to the shape of how NAMS are going to be rolled out will shape European science policy for the next decade. The Commission's roadmap, the Biotechnology Act's, we believe, narrow NAMS definition and growing political pressure to accelerate replacement have created a policy environment that risks, we think, outpacing in many areas the existing science. NAMS, organs on a chip, organoids, advanced computational models do represent significant scientific progress, and EARA 100% supports their development. But in many areas, they're not yet capable of replacing animal models across the breadth of biomedical research. Particularly, I think, in basic and fundamental research, they are nowhere near doing so. So the question for EARA is not whether NAMS should be developed. Of course, they should. The question is whether policy should be built around where the science is, not where advocates wish it were. So EARA's position, NAMS or animal research, is not and, but it's definitely an or. Not an or, should I say. So hopefully most of you will know who King Canute is. So King Canute staged his famous demonstration to prove to his courtiers, that even a king, a super monarch, can hold back the tide. The point really was that there are forces that do not respond to commands. I think scientific readiness is definitely one of them. NAMS are developing fast, as fast as the slide deck. NAMS are developing fast, but they cannot yet replace animal research across many areas of biomedical science. And legislation that says otherwise doesn't really change the fact. Policy should be built around where the science is, not where advocates wish it were. EARA's answer is very clear. NAMS and animal research must advance together. Patients need the full toolkit, as does science. Hopefully today's discussion will help clarify the current moment. So over to you, Nuno.
Thank you, Kirk. I will stop sharing just for a second because apparently you are all seeing my taskbar, which is not what I intended to do. So let's see if it works now. Do you still see my menu?
Yes.
Okay.
That's good.
You do?
No, you don't see.
No, you do not. Okay.
Just
perfect.
a slide.
Okay, so it's my pleasure now to introduce Magdalena Lorenovics. She's head of the Advanced In Vitro Model Systems at the Biomedical Primate Research Center in the Netherlands. And it's my pleasure to present Magdalena, so the floor is yours, Magdalena.
Thank you, Nuno, for the introduction. Indeed, I'm here to represent the Biomedical Primate Research Center in the Netherlands. Next, please. Here I briefly tell you what BPRC is. We are a research institute that conducts exploratory and applied medical research on life-threatening diseases that affect humans. And the purpose of exploratory research is to understand the pathology of disorders that we are studying, which are mostly chronic and infectious diseases. And the purpose of our applied research is to contribute to the development of medicines to the serious disorders. And so at BPRC, we are the largest non-commercial primary research center in Europe, and with the most modern animal well-being standards. And if you would like to look a little bit more how we care about our animals and what we do, you can look at our website. And we are the monkeys that help us understand this diseases that we study are two types of macaques, which are rhesus macaques and long-tail macaques. Next, please. As Nuno introduced me, I'm leading the Advanced In Vitro Model System department, and what we do is we translate clinical questions that are important for humans by developing in vitro disease models, and then there we use both macaque tissues and human tissues to be able to translate what is important for humans in macaques. And then we also learn from our in vivo models in macaque and try to improve our in vitro systems, but also by learning more molecular in molecular way. We can improve our in vivo models, which altogether leads to use of less animals. We study mostly neurodegenerative diseases and infectious diseases and all crossovers between these two. And like that, we uniquely have this very comprehensive platform to study in the disease pathology, but also to develop the new medications. Next, please. I would like to give you here an example of a disease that we are recently studying in the institute in my department, which is post-acute COVID syndrome, or in different words, long COVID. So we all remember still the recent pandemics, but for many people, namely almost 10% of those that were infected, this is still a reality. So they are suffering from for recurring and persistent symptoms. And this long COVID is defined as a disorder when you keep having the symptoms for longer than 3 months. It is very complex disorder, so that's why it's very difficult to understand it and to study it. Patients experience most debilitating the symptoms, cognitive symptoms, so called brain fog. Next, please. And also musculoskeletal system problems, which are connected with fatigue, and digestive tract problems. And at BPRC, we're trying to zoom into these neurological problems and digestive problems, and we think that they are connected. Next, please. So how do we do it? So back in the pandemic times, we have tested over 20 different vaccines, and with one which was eventually successful. But next to testing vaccines, we were also keeping the preserving tissues from these animals to be able to later to analyze them closely and understand better the viral infection. So this is what we do now. We process this and analyze these tissues, but we also kept the animals longer to understand also long-term consequences. And it is not only long COVID that we are looking at, but also really long-term consequences, like what can happen in our brains, for example, over 10 years, because we think that over 10 years we can, due to the overwhelming effects that virus SARS-CoV we can have pandemic of Parkinson or Alzheimer's disease. So with this model, we can both longitudinally take samples, which is quite unique and cannot be done in humans, and also take samples that are
I think we lost Magdalena for a bit. Let's see if she returns quickly. I think Magdalena is back.
Yeah, I was kicked out for some reason.
Oh, so
I don't know what's
probably
happened.
Microsoft. Yeah, you were talking about the why this model we can do is impossible with humans. That's where we
Yes.
were cut off.
Okay. Thank you. Yeah, so we can access the tissues that are very difficult or impossible in humans, and that's what makes this model quite precious. Next, please. But we also generate in vitro models from the tissues of the macaques, and where we can study molecular mechanisms with a bit of less complexity than in a whole organism. But in the same time, within the same species, we can compare these in vitro versus in vivo data. And next to that, we also have in vitro human advanced cultures, which thanks to which we can translate this data even better to human situation. We also generate computational bioinformatic models, and like this, we try to have this holistic approach. Next, please. So I would like to give you an example of how we study the neurological consequences of SARS-CoV-2 infection. And we focus here on extracellular vesicles, which are small bubbles which contain information that is transferred from one cell to another, and this happens in healthy situation, but also in a pathological situation. Next, please. Where the contents of this little bubbles changes. So we can think of them, next please, as small packages with biological information that are transferred from one cell to another, and this information could be RNA, DNA, or proteins. Next, please. And this packages can be released to our blood stream, and from there we can isolate them and read what is happening in our body, in this case in our brain. And so in this invasive way, we have sort of a biomarker which can read what is going wrong. Next, please. We have done first this computational analysis from publicly available data from vesicles derived from patients, SARS-CoV-2 infected patients, and also patients that were affected with neurodegenerative diseases, and we saw a lot overlapping features. Next, please. And there, then we proceeded to do the same in our model, but then before infections and at certain time points during infections. And also here, next please, we have seen that in this little packages we can see the genes that are differentially regulated in the that regulate this neurodegenerative disorders. And now what we do is we take these vesicles from long COVID patients and look at them and see whether we can confirm these signatures. Next, please. Next to that, we model these SARS-CoV-2 infections in different types of brain cells to understand more molecular what is happening, also adding this vesicles and seeing how they affect the brain cells. Next, please. And I would like to finish by summarizing it with this take-home messages. So with a macaque model, we can more holistically look at the disease picture By using parallelly our in vitro brain models, we dive more into molecular mechanisms, and we can exchange information from in vivo to in vitro. And by this, we can have comprehensive understanding of a disease and also start to think about potential therapies. Thank you for your attention.
Thank you so much, Magdalena. That was a beautiful case of combination of both new approach methodologies and animal studies. Now we have Miguel Prudencio. Miguel Prudencio is a principal investigator at the Gulbenkian Institute of Molecular Medicine in Lisbon. The lab is Plasmodium infection and anti-malarial interventions. And the floor is yours, Miguel. Just to remind everyone before, all of the questions that you have, please put them in the Q&A, and we will get back to them at the end of the presentations during the panel discussions. Thank you. Go ahead, Miguel.
Can you just confirm that you can hear me, please?
Yes.
Yeah. Okay, great. So we can start. So first of all, thank you for the invitation to be here today. As Nuno kindly mentioned, I'm a principal investigator at the Gulbenkian Institute for Molecular Medicine and at the Faculty of Medicine of Lisbon. And my lab works on malaria, and specifically our goal is to try to contribute to the development of strategies to combat this infectious disease. If you could move to the first slide, please, Nuno. Yeah, so for those of you that may not be familiar with this disease, let me just outline a few key aspects of it. First of all, this is a parasitic disease that is caused by Plasmodium parasites and transmitted to humans by the bite of an infected Anopheles mosquito. And there are millions of cases of malaria every year, which result in hundreds of thousands of deaths, most of which in children under 5 years old, and with major impacts in the productivity and the public health budget of affected countries. So there are several species of Plasmodium parasites that can cause malaria in humans, but we are most concerned about two of them, Plasmodium falciparum and Plasmodium vivax. If you could move to the next. Yeah, so these are two species of Plasmodium parasites that cause malaria in humans and which have different geographical distributions, different impacts, but either of them has imposes a major burden, and this is why it is important to develop strategies to combat each one of these two parasites that cause malaria, besides other species that cause less severe disease. Can you move to the next slide, please? So just very briefly, this is a very complex parasite with a very complex life cycle, which has to go through two obligatory hosts, the mosquito host and the human host. And within the human host, the parasite goes through several stages, initially in the liver with no symptoms associated, and then subsequently in the blood, where it is responsible for the invasion of and the destruction of red blood cells and for the symptoms of malaria. Next slide, please. So this complexity and of course the burden of this disease means that control measures are necessary and in place to try and minimize the impact of malaria. And these include vector control, treatment, chemoprevention, and vaccination as a means to try and reduce the burden of malaria. Can you go to the next slide, please? So in my lab, we work on several approaches to try and contributing to this fight against malaria. So we investigate potential antiplasmodial compounds that can be used to treat and to prevent the disease. We have long been involved in the development of a novel strategy for vaccination against malaria, which I'll tell you briefly about. And we are also interested in co-infections between Plasmodium parasites and other infectious agents that are prevalent in malaria endemic regions and whose presence in the same host may affect the outcome of disease. So if you go to the next slide, please, I will go straight into the vaccine work that we have been doing, because it is widely accepted that an effective vaccine is crucial to achieve malaria elimination. We now have 2 vaccines licensed by the WHO, but these are not perfect vaccines. Their efficacy is below ideal. And of course, there is much work going on towards developing increasingly efficacious vaccines. And next slide, please. And and of course, studying vaccines and developing vaccines requires access to a whole organism, because we are talking about the immune system and the complex immune responses that are elicited by vaccination, and which involve different organs within their mammalian host, and of course then a very vast plethora of cells, immune cells, that are crucial to mount an immune response and protect against disease. And of course, some of these is simply not accessible in humans, and animal models provide a unique opportunity to investigate vaccines, to preclinically investigate vaccines, next slide please, towards the development of increasingly effective vaccines. With animal models, we can have access to not only antibodies and circulating immune cells, but also to the organs where these cells are produced and where they exert their protective activity, which is obviously something that is very limited and very difficult to do in human subjects. Next slide, please. So this is why during our own work towards the development of a new malaria vaccine, we have resorted to animal models as the sole means to advance into clinical trials. And just very briefly, our proposed strategy for vaccination uses a rodent malaria parasite, a Plasmodium parasite that does not cause disease in humans because its natural host is rodents, and this is called Plasmodium burgii. And we modify this Plasmodium burgii to make it express proteins of the human infective Plasmodium. In other words, we sort of disguise our rodent malaria parasite into a human malaria parasite, and in this way hope to present the immune system, the human immune system, with the tools that it needs to mount a response against the human parasite. So when we proposed this, we had to go through a very complex step of preclinical evaluation of the safety and the efficacy of this product, of this candidate. And this required the use of multiple animal models, which were necessary not only to establish that this would be safe to administer to humans, but also to establish that this product would have the immunogenicity, meaning the potential efficacy that we were looking for. And in fact, if you could go back, please, so just to say that we used several animal models, including non-human primates from the BPRC that we just heard about. And this was what allowed us to go into clinical trials with our candidate and to show in humans that our vaccine candidate was 95% effective in reducing infection by the human Plasmodium in vaccinated individuals, and also allowed us to dissect the immune responses that were elicited by vaccination. Now, can we go to the next one, please? Okay, in the interest of time, I'm moving straight to another topic of interest in our lab, which is the co-infections between Plasmodium and other pathogens, because regions where malaria is endemic are also endemic to many other pathogens, including viruses, other parasites, bacteria, etc. And in the lab, we have studied several co-infections between Plasmodium and these other agents. And today, because I don't have time to tell you everything we've been doing about this, I'm just going to illustrate this with an example. If you go to the next slide, please. An example which is co-infections between Plasmodium, the malaria parasite, and the virus that is called Epstein-Barr virus, which is responsible for endemic Burkitt's lymphoma, which is the most common pediatric cancer in Africa, and this malignancy is strongly associated to the chronic exposure to both the parasite and the EBV virus. And so, next slide, please. We knew from epidemiological studies that the two pathogenic agents coexist in the same regions, as can be seen here, and a very clear correlation between the simultaneous presence of the two and the development of Burkitt's lymphoma has been established. And this thanks to clinical studies that were typically performed retrospectively. However, there are a number of things that we simply cannot access in humans by performing a retrospective clinical or epidemiological study. So, for example, we do not know. So, can you move to the next 2 or 3? Yes, exactly. So, we do not have access to key tissues that are vital to understand the way these two pathogens interact with each other. We cannot know exactly when the subjects were infected with one pathogen and then the other. We don't even know which one came first. We just know that both were detected in these subjects, but we don't know the time of infection by each one of them. And besides, there are a number of confounding factors that cannot be accounted for in studies like this. Next one, please. And finally, of course, there are very severe constraints to the collection of relevant tissues that are necessary to fully understand what is going on in a co-infected subject. This is why animal models provide a unique opportunity to really investigate how these two pathogens interact with each other. And in this particular case, we have been using a mouse model that can be co-infected with murine herpes virus 68, which is akin to the Epstein-Barr virus, and with the rodent malaria parasite, Plasmodium burgii, that I mentioned before. And this tool, this animal model, allows us to go deep into the immunological and other interactions between the two agents that are otherwise not accessible in humans. And for instance, this allowed us to show unequivocally that in a host that is already infected with the virus and is subsequently infected by Plasmodium, the infection by Plasmodium is very significantly decreased compared to a host that has never met the virus. And next slide, please. Conversely, we have shown something very important, which is that so the virus can remain latent for long periods of time and can be reactivated by different factors, not all of which are understood. And our model enabled us to unequivocally show that the malaria parasite can actually reactivate a latent virus, a latent herpes virus, and therefore lead to the symptoms that arise from the viral infection. So we are obviously working to understand the molecular mechanisms behind these, and this can only be done using these animal models that enable us to reach the tissues and to precisely time the two infections and to understand how the two pathogens interact. With this, I would like to finish just by briefly mentioning, please next slide, Nuno, just to say that this is a very timely talk because only 3 days ago was the World Malaria Day, which this end had the motto, now we can, now we must end malaria. And so animal models do play a vital role in trying to devise strategies to combat this devastating disease. Thank you very much.
Thank you, Miguel. This was a very clear example on how complex diseases can get, especially if you're talking about co-infection and two diseases happening at the same time. And now you have to divide strategies to get the best model that you can. And in this case, it is an animal. So next, we have Walter Westerwink. Is a sector of innovation team at the Charles River Lab. And now I give the floor to Walter for his presentation. Thank you so much, Walter.
Thanks for inviting me for this webinar. For people who do not know, Charles River is a global CRO that performs safety services for pharma and chemical industry. And the focus of my own team is the implementation, validation of NAMs and genetic tox assays, and also setting up new test strategies. And today I would like to talk about how we as a contract research lab combine NAMs with animal studies. As a CRO, we evaluate scientific or regulatory questions from our clients, and then we select the most appropriate test system, but more often it's a combination of test system to answer those questions. And the goal is then to assure safety for humans and also for the environment. And our teams, we have different teams. They are trained for in silico, in chemico, in vitro NAMs, but also for in vivo approaches. And the context of use is very important for us to select the best test strategy, because working with NAMs provides us with more detailed mechanistic insights and opportunities for higher throughput early screening. So we need less compound, for example, and we can deselect the optimized compounds. And the animal studies, they give us a better understanding of systemic responses and chronic treatment. The validation and the regulatory acceptance of NAMs, that's ongoing. It's not going very fast at the moment. And animal tests have currently a larger established regulatory frameworks and are therefore often required for global registration of compounds from our clients. And we work, of course, according to the 3 R's as a CRO. Where possible, we will use the NAMs, and if needed, we will perform animal studies. So testing is often a tiered approach. Next slide, please. This slide gives an overview of the current use of the NAMs for the different industries. So NAMs for cytotoxicity, genotoxicity, skin sensitization, eye and skin irritation, they are well established and also broadly accepted for hazard identification or for screening. But the context of use that can differ per industry. For other endpoints, like for example, phototox, endocrine disruption, dermal penetration, NAMs are increasingly used, but the relevance, acceptance, it depends on the industry and the regulatory framework. And the NAMs for the very complex endpoints, like organ toxicity, developmental toxicity, reprotox, they are currently under development and more used for screening and mechanistic testing. And they are in general not yet regulatory accepted. And on the next slide, I will give an example of how we combine NAMs and animal testing for skin sensitization. So a toxicity endpoint where NAMs made a lot of progress, that's skin sensitization. That are, in fact, allergic reactions in the skin. But for that endpoint to give a good answer for all the chemicals, animal testing is also required. As for a significant percentage of chemicals, we need an animal test, and I will explain that later. So for chemicals, we really use a tiered approach. So a skin sensitizer, that's a compound that can induce an allergic reaction in the skin, and that can result in an adverse effect, which is called allergic contact dermatitis. And that's characterized by red, dry, itchy skin, and sometimes you get swelling and blisters. And it's a very important consumer occupational tox endpoint. Traditionally, animal testing was used for this endpoint. One of the tests that was used was the local lymph node test in mice, for example. The test item is then topically applied, and proliferation of the immune cells is then measured in the lymph nodes. But what we do today is a combination of in silico, in chemico, and several in vitro cell culture methods. And animal testing is then more used as a last resort, when the conclusion based on NAMs is not possible. Next slide, please. So the new test strategy that's based on the adverse outcome pathway of skin sensitization that is shown on this slide, and an AOP that's more a structured representation of the biological key events from the chemical exposure and the molecular initiating event to an adverse health effect. And for skin sensitization, there are 4 key events. It starts with the binding of a chemical to skin proteins that then activates keratinocytes that are in the upper layer of the skin. They started to produce cytokines that activates dendritic cells in the skin. They then start to express certain surface markers, and that then finally results in the activation, proliferation of T cells in the lymph nodes. Now, based on a lot of data, it has been shown that assessment of the first 3 key events, that is sufficient to make a good prediction for skin sensitization. So for these 3 key events, a lot of NAMs are available, and they all have their own advantages, disadvantages. And some of the NAMs that are available and offered at Charles River that are indicated here in blue. So the first key event is addressed by the DPRA, kinetic DPRA in the EARA assay. Key event 2 by the keratinosense test. And the third key event is addressed by the HCLAD and the USense test. And in addition, there's an in silico method, in silico DERC, or an OECD toolbox that can give additional information. But not all these NAMs are needed for every compound, and that's why we have implemented a tiered testing approach. Next slide, please. So what we do, we start with a pre-assessment that consists of the in silico assessment. We perform a solubility test. We look at the type of compound. Is it a monoconstituent? Is it a mixture? We look at some physical chemical properties like the log P value. And based on that, we decide what is the best test strategy. That is often one of the test strategies that is described in an OECD guideline, in the OECD test guideline 497. And that the guideline describes how the different assays, the best way to combine them. Then we perform the testing and try to come to a conclusion for skin sensitization. But what we see in practice is that it's not always possible. And in those cases, we need to perform an animal test. Very rarely, we also start directly with an animal test after the pre-assessment. Next slide, please. So how good are the NAMs for skin sensitization, and why is animal testing still needed? So currently, we can come to a conclusion for skin sense with only NAMs for approximately 60% of the chemicals. And animal testing is then used for the compounds that are outside the applicability domain of the NAMs. That are, in fact, the poorly soluble compounds, compounds that have a very high log P value, compounds that give, for example, interference with the assay readout, or have a different mechanism that's not detected with the current NAMs. For example, metals, they have a different skin sensitization mechanism that's not covered by all the NAMs. So currently, only by using a combination of NAMs and animal testing, we can assure safety for all the compounds. Next slide, please. That concludes the example. Now, based on our experience in the CRO setting, both NAMs and animal testing have clear advantages and limitations, and they can complement each other. And this slide summarizes some of these experiences. So NAMs, they offer a high human relevance, provide mechanistic insight, it's very important, and they can allow faster screening with a lower compound demand. And in some cases, they are cheaper. They also support the 3 Rs by enabling partial or full replacement of selected endpoints. However, they also have limitations. They have restricted coverage of systemic and chronic toxicity. There's evolving regulatory acceptance, and there are applicability domain constraints. And one-on-one replacement is often not possible, meaning that multiple NAMs need to be combined, and that can make the science quite complex. And it can, in those cases, also the NAMs can become more expensive than an animal test. Animal testing, in contrast, it captures the integrated whole organism response, has a broad range of endpoints, and it also remains the regulatory standpoints for complex toxicity. You can combine it with more modern technologies like genomics, sequencing, that can be incorporated in the animal test. Limitations are often higher costs, longer timelines, and a species translation between different animal strains, but also between the animals and the humans. And there are, of course, ethical considerations. So in practice, understanding advantages and limitations allows us to select the best test strategies. And it's also good to say that it's really a changing environment. So every year there come new NAMs, and you have to re-evaluate your test strategy. Next slide, please. Then the conclusion and the takeaway message. So both NAMs and animal testing are currently needed to ensure safety for all the compounds. It's very important to clearly define the scientific or regulatory question. And then you can start with NAMs whenever that is possible. In general, they drive human relevance and early decision making. They can already be used for a full replacement for several acute toxicity endpoints like skin, eye irritation, corrosion. For example, also for off-target screening for monoclonal antibodies that have no target, for example, in animals. For other endpoints, we use a hybrid approach at the moment, for example, for skin sensitization, but also for genotoxicity. And the animal studies, they remain essential for complex systemic endpoints where regulatory accepted data are required. The near future of safety assessment lies in the smart integration of NAMs and animal testing. And on the longer term, we expect more gradual shift towards increased use of NAMs. And the final slide. But it's good to realize that using NAMs for safety testing isn't just a one-to-one replacement. It really requires a transformation of how we do science. We have to combine different assays and also to include modeling. And we have to build experience and confidence, and that will take some time. That's the end of my presentation.
Thank you so much, Walter. There was another great example on how complementarity is the word here, and especially with animal testing and the evolving landscape of new approach methodology. Thank you so much. So next we have Sonia Becken. She's the coordinator of non-clinical evaluators at the Belgium Federal Agency for Medicines and Health Products, and she's also the European Medicine Agency chair for the 3Rs Working Party. So Sonia, thank you very much, and the floor is yours.
Thank you very much. Can you hear me?
Yes.
Perfect. Thank you, first of all, for inviting me to give the regulatory perspective on acceptance of 3Rs testing approaches and NAMs. Next slide, please. So I'll just start quickly by giving you a small background on the EU regulatory framework for medicinal products. This is the area I'm specifically working on. We have a directive 2001/83 which governs the requirements for bringing medicinal products to the market, medicinal products for human use, that is. And in this directive, the directive for the protection of animals used for experimental purposes, and hence the 3Rs principles, are completely enshrined. And this is even more so, next click please, with regards to the update of the, thank you. Yeah, the update of the EU pharmaceutical legislation that is currently a final text that is published on the website of the European Commission, where the emphasis on 3Rs is even improved, and where applicants for a marketing authorization application will have to demonstrate compliance with the 3Rs. Now, when looking at medicinal products, we are in a framework where we do not only identify hazard and risk, but we really do benefit risk assessment. And this benefit risk assessment is based on the assessment of the quality, the non-clinical or preclinical data, and the clinical data. And this is to support not only marketing authorizations, but also clinical trial applications spanning from first in human applications where healthy volunteers or patients are included up to phase 3, phase 4 trials. Next slide, please. So with regards to the 3Rs, the European Medicines Agency has been very active already for several years. We had a first expert group on 3Rs set up around 2010, and now since 2022, we have an actual standing 3Rs working party, which I chair. You can find a lot of data on the web pages of the EMA, not only the web page of 3Rs Working Party, but also the web page on the ethical use of animals, which comprises a lot of guidance and reflection documents related to 3Rs and animal use. But also there is a dedicated web page on regulatory acceptance of NAMs. And I'll take you through the highlights of what we've been doing with 3Rs Working Party to foster uptake of NAMs. We had a biannual report also published not long ago, so if you want to consult that, please feel free to scan the QR code. Yeah, next slide, please. So with 3Rs Working Party and EMA, we do not work alone in isolation. We want to collaborate with all our stakeholders to really foster uptake of 3Rs methodologies. And we do this through different kind of interaction mechanisms. First of all, we have 3Rs Working Party, which has different initiatives to engage with stakeholders, such as a stakeholder meeting, consultation on work plans, on all our guidance documents and reflection papers. And as you can see on the bottom of the slide, we have different types of stakeholders comprising not only European agencies, Commission institutions, the European Centre for Validation of Alternative Methods, the European Pharmacopoeia, but also pharma industry, CROs, NGOs and animal welfare organizations. We have also research consortia, 3R centers, etc. So all these stakeholders can interact with the EMA to gain advice on how to progress a particular NAM up to regulatory acceptance, and I'll get back to that in a minute. And we have also interactions through different collaborative fora on European level, such as the European Partnership on Alternatives to Animal Testing. We collaborate with HESI Global, with NC3Rs. We have interactions on scientific meetings and conferences, and of course with the European Commission roadmap for phasing out animal testing for chemicals. And we also took the initiative to set up an international medicines regulators working group on 3Rs, a whole mouthful, to indicate that we are interacting with our sister agencies in Japan, Switzerland, Canada, Australia, and US to try to foster harmonization with regards to acceptance criteria for NAMs, but also exchanging experience with particular NAMs, with qualifications, and everything to achieve progress in the field. Next slide, please. Yeah, just some info. If you scan the QR code, you will see the public session that we held at the last annual stakeholder meeting at the end of March of this year. And you will also find access to an interactive Slido that helps us, in fact, to query our stakeholder for input on our work plan and our activities. So please feel free. The Slido is still open and you can provide your comments. Next slide, please. So let me start with the beginning. We hear a lot about NAMs or new approach methodologies, and we have decided with the 3Rs Working Party to have a fixed definition. What is a NAM for us? NAMs for us are defined as innovative test methods or testing approaches that are aimed to progressively replace, reduce, and refine traditional animal studies. So really enshrining the 3 R's in the definition. These include all types of technologies, in vitro, in silico, in chemico, but also ex vivo systems, refined in vivo systems, and other advanced biology approaches, and structured combinations of these. So with a large emphasis on the integration of all data and weight of evidence approaches. Clearly, NAMs for us are designed to generate data that provide an equivalent or an improved translation to support either human or target species relevant regulatory decision making. Next slide, please. So from the NAMs definition, we move to regulatory acceptance. So this refers to the official recognition by a regulatory authority such as ourselves that a method, a tool, or a data set is scientifically valid and suitable for use in regulatory decision making. So decisions that we're taking within the frame of clinical trial assessment or marketing authorizations. Next click. So this means that for this regulatory decision making, we have to consider the context of use, and this is the specific intended application of the NAM in the development or the regulatory assessment of medicinal products and the conditions under which it can inform regulatory decision making. Next slide. So why is this context of use so important? Because this context of use will drive the type of regulatory acceptance criteria we need to discuss and the extent of them. So clearly there are several building blocks to regulatory acceptance of NAMs, of which context of use and identification of the limitations of a method are important, but we also need to know what is exactly the test methodology, so what are the protocol and the endpoints. We need to have a demonstration of the relevance within that particular context of use, including notions on accuracy. And then finally, reliability and robustness need to be demonstrated. Next slide. And you can click until the slide is complete. Thank you. So the context of use comprises several things. If we are looking on the left-hand side to the development of a drug, we can see that there are different phases and that the non-clinical study requirements follow the clinical development phases with amounting needs for information. And you can see the different type of studies that are usually required for each phase of clinical development. So this can be a context of use. You use a NAM to address safety, pharmacology, genotoxicity, for example, prior to phase 1 or a later phase. But a context of use can also be looked at, and on the right-hand side, you can see the context of use that were developed for organ-on-chip technologies by the ORCID consortium. And you can use a NAM to elaborate disease mechanisms, to test for drug efficacy or safety, and also to use these tools within a personalized medicines application. Now, clearly, as I said, the criteria that underlie regulatory acceptance will be driven by this particular context of use, and we see that the use of NAMs is already diverse. We can see them integrated in weight of evidence approaches, as is done for the assessment of the need for carcinogenicity bioassays, or they can be used as part of integrated testing strategies, such as is done to assess the potential for lethal arrhythmias of a drug, and very limited we are seeing one-to-one replacement. So really this is an option that is rather the exception than the rule. Next slide, please. So to aid applicants and method developers and end users to define context of use and to see also what is the state of the art of 3R's implementation, we compiled 2 inventories on the regulatory testing requirements for human and one for veterinary medicinal products and the opportunities for implementation of the 3R's. And you can see an excerpt on the right-hand side of these inventories where you see the specific topic in column 1, the regulatory provisions in column 2, and these are EMA guidelines as well as guidelines on international scale at the level of the International Conference on Harmonization, but also the Pharmacopoeia, for example. In the third column, you find the animal testing requirements, and in the fourth, the already implemented 3Rs opportunities. In the example that you see here, so the requirements for pyrogenicity testing for injectables, for instance, you will see that there are no more requirements for animal testing, and the Pharmacopoeia fully deleted the animal testing requirement already with a big conference that was also held this year to discuss the alternatives. Next click. So in the realm of non-clinical or preclinical testing, you can see that there are already several indications of possibilities for reduced non-clinical packages, such as is the case for biotechnology-derived medicinal products or products for the treatment of advanced cancer. And often weight of evidence approaches are proposed there for study waiving, such as it is also done for particular endpoints as carcinogenicity testing, juvenile animal study testing for pediatric drugs, and there are also the possibilities to use alternative assays for embryo-fetal toxicity testing, more particularly for the detection of malformation and embryo-fetal lethality. Next slide, please. So how can one achieve regulatory acceptance? And this is in fact a three-step approach, and you find the particular guidelines on the right-hand side. The first and straightforward one is a case-by-case acceptance within a particular product submission. And this is the case for NAMs that are used for regulatory decision making. So in the context of a clinical trial application or a later marketing authorization application. And in that context, the evaluation of the data that are provided on the basis of the NAM will be evaluated on scientific validity or merit and the relevance to the medicinal product and a target indication. So it's very a case-by-case evaluation. Next click. The next one is the EMA qualification for broad use of a NAM, and this is a formal process that exists within the European Medicines Agency and that assesses and endorses a novel method for a particular context of use in the development of a medicinal product. And this qualification will provide regulatory endorsement really for broad application by any applicant wishing to use that NAM, because the whole protocol and SOP will be published on the EMA website. Next click. And then finally, there is the integration of a particular NAM into a guideline or into a monograph or a text of the Pharmacopoeia, which is also possible. And you find some examples on this slide, such as the adoption of the MAT or the bacterial endotoxin test as a replacement for the rabbit pyrogen test. Next slide. But again, I want to reiterate that the requirements are really dependent upon the context of use. And I will give two big examples here. And it's also nice to see it in the context of the previous speakers. So you have two big buckets, whether NAMs are used either for the pivotal safety assessment of a medicinal product or whether they are used to demonstrate the efficacy of a drug within a particular pathology. So looking at risk tolerance, evidence nature, and regulatory expectation and decision focus, this is quite different from one bucket to the other. Now, looking already at regulatory expectations, data that are obtained in a NAM that will lead to pivotal safety decisions will often be subject to ICH guidelines or EMA guidelines, will be thoroughly assessed in product-specific procedures and could be subject to qualification. Whilst looking at whether data are delivered to demonstrate the efficacy of a particular drug will be dependent on a larger evidence base, including clinical efficacy data, and will be relying on best scientific practice and supporting the evaluation of efficacy in the clinic and be often seen as supplementary. And hence, we need a flexible and state-of-the-art approach in that context. So you see here the different kind of requirements, more stringent for pivotal safety assessment, whilst more open and science-based for efficacy. Next slide. Yeah, next slide. So just to recap, and this is my last slide, on the interaction mechanisms that exist at the level of the EMA to gain regulatory acceptance of a NAM. And we are moving from very early advice to stakeholders such as developers of NAMs or end users of NAMs up to the actual acceptance process. So in the first square, you can see two processes that help developers and end users such as pharmaceutical companies or CRO to gain advice. And the very early one is the Innovation Task Force, which is really a forum for early dialogue, and you can exchange informally with the regulatory network on your NAM and see whether the context of use is well defined, what are the preliminary data that you have, what do you need more to gain regulatory acceptance, do you need to go for qualification or not. And important is that this initial meeting is free of charge for all applicants. The next one is portfolio and technology meetings. This is more done with pharmaceutical companies to see also within their product portfolio where NAMs could be integrated. Next click. So the next kind of bucket of interaction mechanisms is more directed towards acceptance of NAM data. And on the first one is really product-specific or portfolio-specific scientific advice. This is formal scientific advice where you can discuss possible acceptance of NAM data, maybe instead of certain animal data, or integrated in a weight of evidence approach gearing towards an adapted drug development program for a particular product or for a particular group of products. The fourth interaction mechanism is the actual qualification of a novel methodology as discussed previously. And then our last option is the safe harbor process. So clearly this is a process to exchange data outside of a product-specific procedure to gain knowledge, to gain trust also with NAM data. This is currently under renovation because we are really decoupling this voluntary data submission from product-specific procedures such as clinical trial applications or marketing authorization applications to make sure that there is no conflict between a product-specific procedure and the assessment of NAM data. And we will be launching calls for data on NAMs within particular context of use within short, where we will be able to appraise the data and see whether they are already mature enough to be discussed for further steps. And next click, please. And I just wanted to draw your attention to the first actual qualification that we have published very recently, where we have qualified the use of virtual control groups instead of concurrent control groups in those range-finding studies to help reduce animal use in medicines development. And this is a first step towards a further process of expanding the concept of virtual control groups for systemic toxicity testing. And with that, I thank you for your attention.
Thank you so much, Sonia. There was a great overview on how advanced the regulatory uptake of NAMs is now in the EU. I would like now to invite all the speakers to turn on their cameras for the panel discussion. A short time, we have around 20 minutes, but we'll try to cover a lot of ground, namely a lot of questions that we got from our Q&A. I would like just to go a little bit back and go ask Kirk specifically about, because we heard a lot about animal research, animal testing. Sometimes those two are clumped together, and the use of NAMs for both are also clumped together. Can you elucidate a little bit about that and how actually NAMs, new approach methodologies, and non-animal methods are actually different definitions, just like Sonia was saying, talking about the EMA definition. Can you elaborate a little bit about those nomenclature and definition challenges?
Yeah, I mean, if you go to any conference these days, everybody has a different slide about the various definitions of what NAMs are. And there's been quite interesting papers have been written, I think, about trying to define, is it new approach methodologies, is it alternative animal methods? I think there are a couple of issues that come from this. I think that one of the issues for regulators about the confusion about what NAMs is, that it risks applying potentially different definitions, which might mean that methods accepted in one jurisdiction and not and rejected in another, which would create inconsistency. I think from a scientific point of view, not having a clear definition of NAMs, it probably leads to kind of funding and validation priorities being distorted. You know, resources will probably flow towards a method that fits the kind of most popular kind of definition, rather than the ones with the strongest scientific case. I think from an industry point of view, I think that companies will face a great deal of uncertainty about which methods satisfy which requirements. And I think from a kind of EARA point of view, the last one I think is probably important, is that if you look at potentially the new Biotech Act, where it defines NAMs as purely replacement, then it kind of over promises to the public what NAMs can actually do, because it sets up a kind of credibility gap, and that people think NAMs just means replacing animals rather than refining or, you know, reducing or complementing the kind of animal models. And I think probably the core problem is that NAMs has become just as much a kind of political term as a scientific one. I think that's why we need a degree of clarity on the matter.
So, and there's this politicization of the definition itself, but then it applies differently if you're talking about like fundamental research, for instance, or if you're trying to apply that to animal testing and to replace some animal testing. One question that we have from the audience, and it's basically for everyone, was if anyone has any advice on specifically on ethic committees, on institutions and animal welfare bodies, how they can better ensure the uptake of NAMs. And this is maybe more for Sonia. How can they better ensure the uptake of NAMs as part of the scientist's toolkit where it's available and appropriate? How can they know more about what is already available?
It's not really within my remit, as we are looking really at approval of medicinal products, as the ethics committees and the whole discussion around the implementation of the 2010-63 falls under the remit of DG Environment. But I'm also part of an ethical committee here in Belgium, and within our initiatives in Belgium, what we've done, we have a kind of matrix for the assessment of experiments, and where all the options of looking at NAM data on triage opportunities are also mentioned. Also, I know that at the European level, there is an annual meeting of the national contact points on the implementation of the directives, where also these issues can be discussed, and there'll be a next NCP meeting in May as well. So if you have issues, please contact your national contact point so that this can be raised at this interface. Thank you.
Thank you. One question for Miguel, and now going back to animal research. One question was, apart from the need from a functional immune system, what else would be needed for an in vitro models to study malaria prevention strategies? Quite a tricky, complicated question.
Well, it does depend on the strategy and what you're trying to develop. So I didn't have time to go into, so I showed you that my lab works on 3 different main aspects of malaria research and of combating malaria. And one of them is the development of or identification of drugs that can combat the parasite. And this we can do we can do a large part of that work using in vitro models like cell lines in vitro systems that enable us to assess the antiplasmodial activity of a given drug. And so this is something that does not necessarily require animal models, at least at the early stages of the process. Then, of course, there comes a point in which drugs need to be tested in terms of their toxicity and in terms of their in vivo efficacy, but a lot of this process can be done in vitro. So the question of what, besides the functioning immune system, it really depends on what we are looking to develop and what we are looking to investigate. If we're looking to investigate a vaccine, it's impossible to do without a functional immune system, and it's also impossible to replicate a functional immune system on a petri dish. So this is a case in which animal models become indispensable. Like for the co-infection studies that I mentioned, there's also only so much that can be done in vitro, and then we need the actual whole system to study the interaction between the two pathogens. But then there are other things that can be done towards combating malaria and towards identifying strategies to combat malaria that do not necessarily require animal models, at least not part of this process does not require animal models. And drug development and drug identification is a good example of something that can be done to a large extent in vitro.
Okay, thank you. For Magdalena, two questions actually. I'll try to make it two in one. So a question is your in vitro samples from the same animals that the ones you studied in vitros? In other words, they are compatible as controls. And do you foresee any advantages of using macaque in vitro models instead of directly using human in vitro models?
So to answer the first part of the questions, yeah, it depends, I would say. So sometimes we do make this the organoids, for example, from the animals that were in the study. This a bit depends on our research questions. Additionally we are expanding our organoids NHP biobank where we can have a plethora of different subjects, let's say, and we could study a little bit of variation between individuals. So basically both options we have possible. The second one, yes, I do, and this is coming from the person who stepped into the primates world relatively recently, meaning 3 years ago, and where so from the point of view of person who mostly did a human study or on human material. So one aspect of it is that many of current NAMs, depending on the definition, so organoid models or IPS, are generated and compared with different species, so human versus mouse, rat. And I think there where the big gap is created, and I think this is a big advantage of comparing within the same species for validation. I think that's unique for when you are working with a non-human primate material. Another aspect is that even though non-human primates are very close and very translatable to humans, there are still small differences. And if you study parallel 2 systems, you can trace these differences, and then you can narrow down if you start testing, for example, a drug, you can narrow down the candidates that you later would like to test in your model. in your in vivo model, which would again reduce the amount of animals that you need, which is again a plus. So here I see the opportunities and advantages of using non-human primates material and NAMs versus human.
Just because this is my question, because I'm curious.
Yes.
Because you're kind of relatively new to the NHP research world, do you feel the increased scrutiny in trying to develop NAMs in light that you are now working with NHPs? Is there an increased pressure, both political, outside pressure, to have these NAMs more readily available, even if you don't think it's necessarily doable at this time?
Yes, the pressure is definitely there. Yes, from all kinds of angles, and basically we were pushed to destiny our funding for the institute for development of NAMs. And I think it's not a good direction because, again, as many already have said in this webinar, this should be two parallel paths. They are complementing each other, and we should learn from the model. So it is really depending on your scientific questions which model you will be using, and as any model, it's just a model. So you cannot say that a NAM is perfect for something just because it is NAM. It needs to answer your scientific questions. So this is for me a worrying direction that animal models are being sort of pushed aside as not being correct anymore. But of course, the new technologies are interesting and important, and will bring us new valuable answers. But we still are not there to connect organs, for example, in a proper way, like we can in animals. So that's my take on it.
Sonia, do you have a comment on that?
Yeah, I have a comment because very recently we published a reflection paper on the use of non-human primates for testing of human medicinal products, not only due to the 3 R's aspects, also related to the shortage of non-human primates during the COVID period. And we have made a lot of recommendations in that reflection paper that is available on our website, but clearly there is an overarching recommendation that NHPs should be used only as a last resort, meaning that all other available sources of information should be used first, and then there should be an evaluation of whether first the NHP is really the only possible last resort that you need to answer the question that you want to see answered. And also, there is a lot of push to include weight of evidence approaches, integrating available data, knowledge on the target, off-target effects, and NAM-generated data. So it's clearly a complementary to what has just been discussed, and maybe it would be good to see how these lines of thinking could be used together.
So to Walter, as a CRO, do you also feel this kind of pressure to move more towards NAM in a quicker way? And there was also a question to you. Let me just get to it, because there are a lot of them. Regarding because you talk about one of the advantages of NAMs was the human relevance, and what does actually, this is the question, how does high human relevance mean actually? Relevance for who? Do these NAMs also represent it was a woman asking, so me, a female non-Western ethnicity. Is there also a worry in getting these NAMs that are human relevant across the board for all people?
Yeah, it really depends on the tox endpoint, what it means. I mean, for acute skin tox, for example, a very general model can be used. That has been shown with a lot of data. But for other endpoints, like for example, liver toxicity, when you do that, for example, with genomics, then you need certainly a mixture of different donors to have a good representation. So it depends on the endpoint. And as a CRO, we also see the pressure to move to the NAMs. But yeah, that's why we use a tiered approach like what I showed in my presentation. Where possible, we use the NAMs. If it's not possible, and that's still the case in a lot of situations, because most of our clients want to register their compounds globally, and the NAMs are not accepted everywhere in the world at the moment. So that's also often a barrier for using the NAMs.
Mm-hmm. So I would just like to go back to a little. We don't have much time. I have to. This is for everyone to comment, because we are seeing more and more NAMs being developed for replacing animal testing, replacing quote unquote, or complementing animal testing. How do you see NAMs complementing research itself? So animal research, how do you see the NAMs complementing not only the testing and the regulatory part of accepting a new therapy, a new medicine, but also to study the fundamental biological processes of that you usually need a whole organism for?
Well, I think that again, NAMs are perfect for certain scientific questions, and I think definitely if we would look at the relevance for human, there were they are probably most translatable, let's say. Again, you need to be caution about that, because they are representing only part of a complicated system. So probably in a couple of years' time, we'll be more going to even more complex system. But again, then when you introduce to these NAMs more complexity, it also brings a more complexity to interpretation of your data. So I think there is definitely a potential, but I think every model needs to be very carefully designed for the questions you need to answer. This would be my take away from this question, at least from my point of view.
Anyone else wants to chime in? Miguel?
I can complement this just to say that obviously, if we can find alternatives that can answer the questions that we need to answer scientifically, then of course those would be very welcome. I think that some aspects of the biology, and particularly in my research, infectious disease research, and some aspects of the biology of host pathogen interactions can be studied using NAMs, but some will always remain very difficult to access in the absence of a whole functioning organism, because interactions are very complex between different types of cells, between different systems within an organism. And so I think some fundamental biology questions can already and will continue to be addressable using NAMs, but some will remain difficult to answer without the use of proper of adequate animal models, or ultimately, of course, humans themselves, and access to human samples and clinical trials that can provide the material that we need to answer those questions. But of course, there are things that we cannot access in humans, and there are biological phenomena whose complexity is not replicable in an in vitro system, at least not for the foreseeable future. And I think this will remain a fundamental aspect of research, of fundamental research, to have access to complete systems where we can look at these issues as a whole and not as discrete systems that don't interact with each other.
Kirk?
Yeah, I mean, a couple of quick points. I think that obviously within basic and fundamental research, where the object is to better understand a kind of a journey of discovery, it's hard to imagine that a new approach methodology, when you're not completely sure what you're looking for, can replace the animal model. So I think clearly within whole areas of basic and fundamental research, then I can't see very quickly how NAMs can replace something you're not even too sure what you're looking for. I think back quickly to the definitional confusion. I mean, I think this is not really a side issue. I mean, until we agree what NAMs are, then we can't really have an honest assessment of what they can do and what they can't do. And if policymakers and regulators and scientists are not working from the same definition, then phase out timetables become often fairly arbitrary. So getting the definition right isn't really just a technical issue. It's the precondition, I think, for a credible science policy.
Sonia?
Yeah, I would like to maybe angle it a bit differently. We are discussing the impact of a definition, but shouldn't we be discussing what is the best scientific way to approach a question, or whether it be a fundamental research question or an applied research question, and whether some tools that we have at our disposal, which are sometimes very innovative tools like in silico prediction models or some very highly complex in vitro models, can give us maybe one part of a mechanistic answer that can help us one step further. And maybe we should see it as an integration of these novel, innovative tools of gaining answers and seeing where do we need maybe an animal model or human to provide an answer or to complete that answer, rather than saying it's this definition or another, because in the end we are only discussing the same thing over and over again.
I'll take this opportunity, just last question, because we're finishing, because there are a lot of work going on right now in the development of these new technologies, and this is probably for you again, Sonia, sorry. How do these developments all over the world internationally can help the harmonization in a way that will improve the uptake of NAMs regulatorily?
Yeah, well, there is a lot of crosstalk between global regulatory agencies, but also including pharmaceutical industry. If we're talking about the pharma, first of all, at the level of the International Conference on Harmonization, there is now the ongoing initiative to draft a reflection paper on NAMs and weight of evidence approaches, and see how we can foster global harmonization in that field. We have also, or we are in the process of having a 3Rs related statement to the International Coalition of Medicines Regulatory Agencies, which is even broader than ICH, to also bring that to the attention and to see where we can foster progress there. Now, at the European level, we have, of course, cross-sectorial, the European Commission roadmap that will be published within soon, that also fostered cross-sectorial harmonization in the approaches. So there is a lot ongoing. We are also regularly discussing with our colleagues across the pond, and hopefully we can make the push together. There is certainly a lot of noses in the more or less the same direction. The implementation is sometimes lagging behind, but we're doing our best to have this dialogue. Yeah.
Okay. Unfortunately, it's time to wrap up this meeting. I would like to thank all of our speakers dearly for their interventions and for their answers during the panel discussion. I would just like to encourage you to fill out this evaluation survey on this webinar, on our webinar series. We will send you a certificate along with this link. And we also, again, would like you to encourage to register for board for our social media campaign that will be thematic this year and will approach new approach methodologies and animals studies in complementarity. So please check our website for this. And again, thank you very much, everyone, for this wonderful session. And so our regular webinars will come back after the summer. In the meantime, I'm happy to announce that we'll be doing an extra webinar with ECLAS very, very soon, and that will be announced in the next few weeks. So again, thank you all, and see you soon.